Hello, good afternoon and welcome back to StudentSOS. Or if you're new here, then very good welcome to you. It's lovely to see you this afternoon. I hope you're all doing really well. So this afternoon I'd like to address something that I wasn't going to look at yet in the AS series, but something that happened a week or so ago at a festival in the United States meant that I decided to try and address this at least briefly here now. So we won't discuss the gentleman involved in what went on, and most of you will know exactly who I'm talking about when I tell you what I'm going to talk to you about today. Because part of the AS level syllabus is to know about the immune system, and a specific aspect of this that you have to know about is HIV virus. Now, HIV is the virus that causes AIDS. HIV stands for Human Immunodeficiency Virus, which if you are an A-level candidate, hopefully you know already. So this is the infectious agent, HIV, and AIDS is acquired immune deficiency syndrome. This is the disease, if you like, the condition that the virus eventually causes. So they are two different things, but they are linked, which is why when you see material talking about this, it's often written as HIV/AIDS. So they are not the exact same thing. So that's an important place to begin. So I'm not going to go into lots and lots of detail about the virus structure today. I will do a video on that at a later stage. But what I'm going to tell you is it's a retrovirus. For those of you who know anything about this, it means that their nucleic acid, their genetic material, is RNA, not DNA. And it's housed inside a protein coat, which is pretty standard for viruses. In fact, those of you who've been keeping up to date with things that have gone on during the pandemic will know quite a lot more than you ever dreamt you want to know about viruses now compared to 18 months ago. So it's a virus inside, so it's a protein coat, a protein coat with a RNA nucleic acid in the centre. It also carries with it a really special enzyme called reverse transcriptase. Now, reverse transcriptase has now been used by scientists and research scientists for many years thanks to the discovery of it within HIV virus. So this has become really useful. Basically, reverse transcriptase is responsible once HIV's nucleic acid gets inside a host cell, then it's able to use reverse transcriptase to take RNA back to DNA. Now, to many of you, this probably doesn't sound like that big of a deal, and to some of you, it will have just gone flying over your heads. Stay with me, I'll try and explain this very briefly now. So what we believed until relatively recently, 30 years ago, was that DNA coded for RNA, which coded for proteins. And there's some videos coming up next week on transcription and translation, which will look at exactly that flow from DNA to RNA to protein. So that was called the central dogma of molecular biology and had been around since the 1960s. So HIV turned this literally on its head because it had an enzyme within it that allowed the RNA that it carried, its instruction manual, to be translated back or reverse transcribed, as per the name of the enzyme, back to DNA. That was super important for HIV. It was the key to it being able to fulfill its life cycle within humans. Because we have DNA as a nucleic acid, not RNA. And what happens is that when HIV infects humans, it goes and travels to white blood cells, very specifically, in fact, what we call CD4 positive white blood cells. So you have to have a receptor on the surface of your white blood cell, which allows HIV entry. You will have almost certainly heard far too much about spike proteins in the last 18 months. Most viruses have these sorts of spike proteins, but they are their attachment mechanism. They are how they gain entry into cells. And HIV is no different in that regard. So these infect white blood cells containing a CD4 receptor on the surface, and on this spike protein they have a spike protein labelled GP120. For AS biologists, GP stands for glycoprotein. you need to know what they are if you don't already. When we take a little look at the fluid mosaic model, we'll take a little look at glycoproteins, but GP120 for everybody else. And this is a specific interaction. This GP120 fits into CD4 receptor and allows HIV virus to gain access to the white blood cell. Now, the most remarkable thing about viruses is they are not cells. Even more remarkably, and almost unbelievably based on what we're currently living through, they are not living organisms. Yeah, I know. They're not living organisms. If you think about that, it kind of makes some sense, because viruses are only a problem to our cells once they're inside them. They don't survive very long on surfaces. It's why numbers tend to fall over the summer. They did last summer, and they appear to, certainly in terms of transmission, be doing so more this year. But nevertheless, viruses generally, like the flu virus, aren't able to be passed on easily from surfaces and things like this, because the virus is destroyed. It's not killed because it wasn't alive. The virus is destroyed really quickly in UV light. That's common to lots of viruses. So flu virus, definitely that's true for. That's why we tend to get them during the winter and the colder months of the year, when we have windows closed, when we're in closer proximity with one another indoors for longer periods of time, and indeed when the weather is cooler. So therefore, if virus ends up on door handles, on tables, on packing surfaces, then it's likely to survive longer, so that if you touch it an hour or two later and then manage to put it inside your body somehow, it's very simple to do. You're going to touch your face once you've touched a surface before washing your hands in between, and then you'll be infected. So HIV is infectious in a very different way, but ultimately viruses are not alive if they are not able to get into your cells. So this is what makes viruses quite interesting from a scientific point of view, and indeed quite terrifying, I would suggest, at the same time in many ways. So HIV virus gets into these white blood cells and it converts its RNA to DNA, and that DNA then actually cuts open your DNA, human DNA, within that white blood cell, and it pops itself in there for a little sleep. And this is called latency. So the DNA from the HIV virus is basically integrated into your own DNA, and there it goes for a little nap. Now, this is one of the reasons why infection with HIV most certainly does not cause death within 2 to 3 weeks. Let me just say that again in case a particular gentleman is watching. HIV infection does not cause death of humans within 2 to 3 weeks of infection, ever. It's extremely important that we know this, because HIV virus tends to stay latent in the white blood cells for sometimes many, many years. When this first was discovered in the early mid-1980s, it was often 15 or 20 years before people developed any real symptoms. And even now, with some of the more aggressive viral variants, you'll have definitely heard the word variant in the last 12 months, we are still talking about 6 to 10 years between being infected and developing symptoms. Occasionally people note a few months after being infected that they have like flu-like symptoms. The problem is people have colds and flus all the time. How on earth would you realise that it was as a result of HIV infection? So unless you considered yourself to have exposed yourself to high risk in a number of different categories, having a bit of flu, whilst unpleasant and a little inconvenient, isn't likely to make you suddenly think you have HIV. Quite rightly, it's very unlikely to be the case. So we have HIV infection that's latent. Now, of course, initially treatments were not very sophisticated. And what we've always used is what we call a multi-combination therapy system, whereby we try and attack the life cycle of HIV at a number of different points. This tends to give us the best opportunity to stop the HIV virus in its tracks. Because what basically happens is once latency is broken, and there's lots of research that's gone on over the last 30 so years to suggest that latency is broken for a number of different reasons, but once latency is broken and the DNA is transcribed, so copied into mRNA, and then translated, that means converted to protein. What now can happen is that virus particles or virions are produced. As they leave the cell, they tend to have upwards of 100,000 virus particles that are released from one single white blood cell. And in so doing, what many of them tend to do is burst the cell open. This is called lysis, for those of you who are not familiar. They burst the cell open and they take with them part of your cell surface membrane, your plasma membrane, and they take it as a jacket around the outside of themselves. This is a remarkably skillful thing to do because now they effectively evade the rest of the immune system whilst they are going to reinfect more white blood cells. Because of course the immune system identifies foreign antigen, which would be something like GP120 on the surface of the HIV as one of its spike proteins. But unfortunately, with this cell surface membrane over the top of it, it's not going to be seen. The spike proteins won't be seen, and so that all that any immune surveillance cells will see is cell surface membrane, human cell surface membrane, your cell surface membrane specifically, and it will ignore it because it's self, it's yours. We don't want the immune system to attack that. That happens very rarely in autoimmune diseases. More on that later. So it's really quite clever because then of course it gets rid of its jacket, HIV, finds itself a new cell with a CD4 receptor, again these are white blood cells, and attaches, in it goes, latency again, fantastic for HIV, deadly to us. Now over a lengthy period of time, whether you are being treated for HIV or not, and the length of time that it will take to get to this next stage will depend firstly on whether you're being treated, which will depend on whether you know you're HIV positive, of course, because you may not, and indeed on how successful that treatment is being with your particular HIV. So it's important to keep in mind, sorry, itchy nose, it's important to keep in mind that there'll be lots of variants of HIV because in common with all viruses, including the virus we're currently struggling with globally, they will mutate very quickly. In fact, I read some research a few years ago that suggests that if you're infected with one single variant of HIV, within 12 months, you can expect to be able to find in the blood plasma over 200 genetically distinct variants of HIV. Now, who cares? Well, that's a great question. The reason it's important is because some treatments will be more effective against certain genetic variants than others. And this is why we use combination therapy. Now, initially, combination therapy was hugely problematic for people because some of the side effects were reported to be so bad that some HIV positive patients chose not to take the treatment because the side effects were worse. Now, this is thankfully not so much the case anymore. HIV is extremely treatable and people go on to live very long and indeed healthy lives with HIV. There's a huge number of reasons for that. We also have drugs that pregnant women can take to reduce the risk of them passing HIV if they are HIV positive onto their unborn child. And so treatments have come a long, long way in the last 30 or so years. So if your white blood cell count falls below a certain level, which can be monitored whilst you're being treated for HIV, then you will develop AIDS. Now, the real problem with AIDS is that you can't directly die of AIDS. So what happens is that AIDS is when your immune system has been significantly weakened, and once that happens, you are now prone to what we call opportunistic infections. So AIDS itself doesn't directly kill you. It reduces the quality and power of your immune system to such an extent that a cold could become pneumonia. There's a very specific type of skin cancer that people with AIDS develop called Kaposi's sarcoma. So there are all kinds of opportunistic infections that may not either occur in people who are not HIV positive, or indeed may not kill them if it does occur in people that are not HIV positive. So AIDS itself simply causes a significantly weakened immune system because, of course, the HIV particles, as they burst out of white blood cells, destroy the white blood cell. And then they go on and infect a load more white blood cells. Now, we can keep producing white blood cells, but if you imagine 100,000 virus particles being released from every single cell that gets infected, this just becomes a game of cat and mouse all the way through. But the treatments clearly demonstrate that in many cases science wins, which is great because then patients win. So overall, what I want you to take away from this is HIV isn't necessarily anymore the same sort of death sentence that it definitely was seen to be in the mid-1980s. I was a teenager when the Don't Die of Ignorance campaign was around, which if you've never seen that, I'm sure there'll be a video of it on YouTube. Terrifying stuff. So really important to know that along with lots of diseases actually nowadays, HIV positive status is not a death sentence for people. So that's extremely important for you to be aware of, never mind 2 to 3 weeks. It's just not the same problem that it was previously because science has come such a long way that patients are able to live very well for an extended length of time with HIV, which is great news. Now, of course, there's lots of ways to prevent infection with HIV, and when we look at the A-level biology material on this, we'll take a look at how that works. But it's a blood-borne disease, so you need to have body fluid contact. So thankfully overall this is a much more tricky, believe it or not, disease to contract than something like coronavirus. So all you've got to do is be in the same airspace as someone who's sneezing droplets all across the room to contract coronavirus in theory. Unfortunately, HIV may have much more significant implications for a greater number of people, but the reality of the situation is that you need to make sure that from a HIV point of view, that you are preventing body fluid contact between you and someone who's infected. I think that overall that's much more preventable in most cases than the situation we're currently in. If you have any questions on HIV, including the gentleman who was speaking so out of turn last week at the festival, if you find me, drop me a comment down below. I'm happy to answer your questions. I gather he's been fully educated on Instagram, which is excellent news. But please, if you have comments about this video or anything else you'd like to know about this topic, please drop the comment below of this video. I'm going to see you in the next video. In the meantime, if you enjoyed this content or found it useful, please hit that like button and hit subscribe if you'd like to be notified of new content. We're releasing new content every single week. It will cover from 11 plus all the way through to UCAT preparation for potential medics. We are science-based from GCSE and A-level. And so if you'd like to get notified of more content as we produce it, please hit that subscribe button. Also, please consider taking a look at our Patreon account. We have a system that's set up where you can access exclusive content, exclusive lectures, and also get discounts on one-to-one work that you would like to do with us. So please take a look at that. Details of that will be down below. Keep your eyes out because StudentSOS.co.uk has been revamped as a website and it will go live in the next week. So please keep an eye out for that. There'll be a video confirming that's released. And otherwise, have a great day. Very good luck to anybody getting exam results this week. Thanks very much for watching and I'll see you next time. Bye-bye.